So I'd now like to introduce our keynote speaker, Kip Guy, who's the Dean of the College of Pharmacy of the University of Kentucky, and also professor in the Department of Pharmaceutical Sciences. Today he'll be speaking to us about discovery and development of SJ733, a novel anti-malarial drug. Dr. Guy obtained his BA in chemistry from Reed College in Portland, Oregon, and his PhD in organic chemistry from the Scripps Research Institute in La Jolla. While at Scripps, he also carried out additional training in physiology at the Woods Hole Research Institute. After completing a postdoc in cellular biochemistry at Texas Southwestern, Dr. Guy joined UCSF in 1998 as an assistant professor with joint appointments in pharmaceutical chemistry and cellular and molecular pharmacology. And then in 2005, he was promoted to full professor. And while at UCSF, Dr. Guy founded two centers that ultimately became the Small Molecule Discovery Center. In 2005, he was recruited to St. Jude Children's Research Hospital in Memphis to found and chair the new Department of Chemical Biology and Therapeutics, where he was the Robert J. Aldrich Chair in Chemical Biology and Therapeutics. He currently holds adjunct academic positions at UCSF, the University of Tennessee, and Vanderbilt. His research is focused on the discovery and development of novel small molecules that target orphan pediatric diseases, particularly pediatric oncology and protozoal infectious diseases. Most of his group's work falls into the area of chemical validation of novel targets, discovery and optimization of novel chemicals for validated disease targets, and the use of whole cell strategies for lead discovery and optimization. He's the author of 167 papers and book chapters, the inventor on 8 issued patents, and is co-PI on 2 NIH grants, one involving the validation of new anti-malarial drugs and another involving novel therapeutics for leishmaniasis. We're honored to have Dr. Guy here today.
For those people in my college worrying, those adjunct appointments are all expiring this year. So I'm fully blue at this point. Okay, so what I want to do is give you a story about translating all the way from an idea up to the clinic. We just obtained funding for this project to move into phase 2. So it is still continuing, although those of you who do clinical development will know phase 2 is the true valley of death, where most new drugs wind up becoming desired products instead of real products. So everyone think positively about this. So this project started almost 15 years ago now out of my laboratory. It's been run and organized by me, but at this point involves collaborators on every continent except Antarctica, and every sector you can imagine, not-for-profit, for-profit, many different academic collaborators. At the end, I'll show you a slide with everyone, and no one will be able to read it, even those right in the very front of the room. So I did want to point out there's several people in the audience who have been intimately involved in this project. So can I ask you guys to stand for a second, just so I can say thanks? Thank you. All right, so let me tell you a little bit about malaria and why we were doing this. And I'll use this to show you how really the clinical needs drive what's being done at the bench and how we return back to the clinic based on that interaction. So this is a disease where roughly 3 billion people are at risk. So anyone in a tropical or semi-tropical country can get this disease. We had it in the US and Europe up until the 1940s to 1970s, depending on which particular part of those continents you're in. And in fact, we still have the mosquitoes that carry the parasite that causes malaria all through the US. So global warming may have other impacts at some point. There are roughly 200 million cases per year, so it's a very high morbidity disease. As of the 2013 World Malaria Report, it caused 630,000 deaths in that year. This number has been trending downwards since the early 2000s. It peaked at about 1.2 million deaths per year. We're down. This year it's projected to be about 450,000 or so, although those numbers are fairly soft. So 80% of the patients who die from malaria are children who are under the age of 5. The majority of these are between 18 months and 3 years of age, where they've come off of maternal immunity and haven't yet reestablished the partial immunity that you can get from repeated re-exposure to the organism that causes the disease. This accounts for 20% of childhood deaths in Africa, all by itself. And that means there's a child dying roughly every minute, every day from malaria. So these are the kind of motivators that keep me going for this disease. I'm deeply motivated by health disparities and diseases of children. If you've never taken a parasitology course or you've forgotten your course, I will walk you through the life cycle. So it's a fairly complicated life cycle. The parasite that causes it, Plasmodium, is vectored in between humans by an insect, the Anopheles mosquito. This is done by the female mosquito when she takes a blood meal to allow her to go through her life cycle. If she's infected with the parasite, it will be in her salivary glands in a form which is specialized for uptake into humans. And it will then migrate from the bite site to the liver, enter a hepatocyte in the liver, and undergo one life cycle here. So this is the sporozoite form, goes into the liver. As it comes out of the life cycle in the liver, it's differentiated into a second form called the merozoite, which is specialized for uptake and reproduction in human erythrocytes. So the patent disease, the symptomology that we associate with malaria, comes from this section of the life cycle. The parasite, you might have, say, 1,000 at a bite, and this can amplify up to 10 to the 10th to 10 to the 12th parasite load with an individual patient. This is an enormous clonal expansion. So that part of the disease life cycle and the dynamics are a lot like a bacterial infection. Any kind of genetic diversity that's within that seeding population that gives any advantage to the parasite at all can be amplified. So imagine, for example, you're carrying an allele that confers resistance to a drug. Right? It will, within a single patient, be able to take advantage of that allele. And then horizontally transmit at that percentage, now amplified because of the selection pressure within the patient. So this is actually the major Achilles heel in drug discovery for malaria. Good drugs last a decade in the clinic as a monotherapy. We don't actually use monotherapies anymore. Many drugs, things that want to be drugs, don't even make it through clinical trials before there's substantial resistance within the population. So we study falciparum. This is the highest mortality of the causing of the 5 species of malaria that can infect humans. It ejects itself, as we understand, completely from the liver after the liver section of the life cycle, moves on to this. But there are other subspecies, Vivax in particular, which can enter a quiescent state in the liver typically for months, but they're documented case studies of up to 30 years post-primary infection. At any time after that, entering that quiescent state, it can come back out and do what's called recrudescent infection, so reestablish an infection without a need for another mosquito bite. So anything you do with drug discovery and development for this disease, you have to deal with the fact that a certain percentage of your patients walking around have a quiescent activatable disease, and which you're probably not able to clear out of that patient. We have no drugs really that work on the hypnozoite, the quiescent form of the parasite. So you have to think about it as a population health problem from the get-go, in addition to caring for an acutely ill patient who can die from the disease. So while it's going through this life cycle, this is relatively synchronized and roughly 48 to 72 hour life cycle. So the waxing and waning fever and the waxing and waning anemia and the other major symptoms that we see in patients are associated with that cycle. While it's going through this at a random level, it can differentiate now into a sexually differentiated form, which is specialized for uptake back into the mosquito. So large asexual reproductive cycle going on within the patient, move into the mosquito after that mosquito feeds on an infected individual, can undergo sexual reproduction within the mosquito, able to generate all the kinds of genetic diversity we associate with that sort of reproductive life cycle outside of the effect of the drugs, right? And then reinfect. So not only can we amplify alleles that confer resistance quite readily, but we can actually generate them quite well relative to a lot of other infectious diseases. So as you might imagine, resistance becomes the underpinning of most programs. So I'll illustrate that with this slide. When we started this particular part of this program in about 2000, there was a new therapy coming on the market that was based on a natural product-derived drug called artemisinin. There are about 5 drugs on market. They're all combination therapies, bundled up in a formulation for a single dose. So I mentioned that the death rate had dropped from about 1 million to about 400 to 500,000. This was because of the introduction of the ACTs. As they've come onto the market, they've had a major impact on morbidity. Coartem was the first of these to come out. It's a Novartis product, and it drove a drop of almost 45% in mortality over the decade that it was out. And what happened was everybody who was doing drug discovery said, this is great, it's combination therapy, it's really fast acting, we're never gonna have resistance. Let's move on to doing things like getting less toxic drugs to be the combination partners to complement the ozonide. And of course, we were fooling ourselves. So roughly a decade after the ACTs came out, in Cambodia, we started to see patients who were responding more slowly to the ACTs, in particular to the AS, which was used there. So we started to worry about this, but not terribly much because it was in a single locus in the world. It's now spread to 5 Central Asian countries, and we're seeing 40% of patients in Cambodia who now actually have patent resistance to ACT. So they're not only more slowly responding, but they're failing in therapy there. So this has moved in a decade from we're not really worried about it to we need to replace the A and the ACTs in order to keep being able to maintain mortality where it is at just 500,000 deaths a year. So this shifted everybody back to focusing on ozonide replacements. And as I walk you through the pharmacology of what this means, you'll see that this makes you a little schizophrenic if you're actually trying to develop malaria drugs. So I will say the constant need, and one of the things that's motivated us as scientists, has been new mechanisms. This is what allows you to evade resistance that exists in the field. If you're thoughtful about the mechanisms and the way you combine drugs of different mechanism, you can slow resistance acquisition to the drug products. All right, so let's talk a little bit about what we're trying to do in the clinic. So the aspirational goal for malaria is called a SIRCAP drug. This stands for single exposure, radical cure, and prophylaxis. So let me unpack that for you. That means this is a drug where we give a patient a single pill. It's gotta be a pill, it's an oral requirement. We cure the disease that they have, including the liver stage. We prevent them from transmitting whatever disease they have while that's going on. And the same drug is usable in a local population, say at a village level, as a prophylactic. In addition to that, which is aimed at the disease endemic country, if we want to recoup research costs in order to enable this product actually getting deployed, we need to be able to sell it to people like this audience who are going to travel from a first world country to a disease endemic country who want to use this drug as a purely prophylactic medication. So there are multiple issues that arise from this. So the single dose in and of itself is a problem. I would ask you to think about how many antibiotics you know that you can give a single dose. I will answer that for you. There are no antibiotics that you give as a single dose. There are a few that you can give as 3 doses. So this is a pretty high bar from an infectious disease standpoint. It's got to kill all of the life cycle stages. So imagine this from a physiology perspective. You got a parasite that lives in an insect and is moving into the lymph system, circulating around there, getting into the circulatory system. You have a parasite which is specialized for living inside a hepatocyte, high oxidative load, all those issues. And then the same beast specialized for living inside of an erythrocyte, basically a metabolically inert, fully cloaked state. Lots of different physiology you got to deal with there. You need to kill 5 different species with a single drug. So again, just think about the antibiotic utility. How many antibiotics do we have that have that kind of spectrum that we use regularly? We'd like the drug itself not to have a high propensity for getting resistance. We'd like, say, a decade of utility in the field, and we'd like this post-treatment prophylaxis. So think about this as we work it backwards to what we need to see in patients and in the laboratory. So effectively, we need to be able to clear 6 logs of parasite load in a patient in order to take somebody who might be close to death back to a level of symptomology where they're manageable as we soak up the rest of the parasites that are present. We need that to happen quickly because these patients have high acuity when they come into the clinic. They're very ill. And at the same time, we're treating a lot of people who are subsymptomatic. We want the same drug to be usable in both settings. Because of the prophylaxis requirement, we needed to have a long duration of action. So we needed to work really quickly, but then hang around for a long time to prevent the bug from moving around. We'll take one or the other of those in a single active ingredient because it's going to be a combination medicine. As I mentioned, it's got to kill the sexual and the liver stages of the parasite ideally. And I also mentioned we really don't have any good liver drugs. We have a few things in mid-development now that might work for that. These are add-ons in any single active ingredient as long as I combine things in the drug that bring them to the table. It's got to be very potent. So think about a double-lock capsule, which is probably the biggest most of you are comfortable in swallowing. That holds about 200 to 300 milligrams of total material for a typical kind of formulated drug. Most people can't swallow more than 4 double-lock capsules in a row, right? And you think about the last time you took tetracycline, for example, it's in that kind of a packaging setup, and nobody likes doing that. So this is a pretty generous number, a gram of total drug product in the dose. And it's got to be safe. Really, really, really safe. We're giving this to young children. We're giving it to women who are pregnant, who don't know that they're pregnant because they're in medical systems where that's not easy to monitor. We're giving it to first world travelers who will get indigestion and think that's a serious adverse event. It's a really wide range of safety and tolerability issues that we have to deal with. That's why I like this area. It's really hard. It keeps you on your toes. All right, I'm not going to bore you with all the logical hypothesis-based science that we use to try to get this project going over a decade because none of it worked. And I was mentioned as somebody who does a lot of cell-based approaches to drug discovery now. This was the project that moved us into that paradigm because out of desperation, we needed to find other ways to identify leads. I hope I'll convince you as part of this talk it works. So we identified a strategy we called phenotypic, where we wanted to look at living or death of the parasite as a primary outcome. And we simply wanted to ask the question of whether or not, while the parasite was cycling in red blood cells, we could kill it effectively without being harmful to mammalian cells. So this is about as simple a question as you can ask to meet all those goals I had outlined for a new drug product. So we set this up as a requirement. It had to work on all the existing drug-resistant strains. It had to be reasonably potent. This is about micromolar and cell assays, and it had to be safe on mammalian cells. This was how we did initial screening. Then we set up, as we were following up on early hits, to exclude things that worked on known mechanisms because we didn't want to get back into an existing resistance manifold. And we actually did this by looking for differential activity against drug-resistant strains of malaria and basically throwing out anything that had a significant differential. Then we would focus strategically on things that had desired pharmacodynamics. They were quickly acting, they were cidal, and they killed a lot of parasites. And pharmacokinetics, that we thought they would be stable in vivo. We thought they had properties that would lead to oral bioavailability. And only at the very end would we deal with identifying targets and thinking about things in a mechanistic sense in the parasite. So I'll try to take you through all of that in the remaining time. So in terms of this project, we were doing this while at St. Jude. We screened, at that time, the whole collection. It was about 300,000 molecules in a cellular assay where we co-cultured the parasite in red blood cells. And we asked whether or not the parasite evaporated at a pretty high dose, a 7 micromolar fixed dose. And I've thrown in on the slides dates so you can see when things happened and papers if you want to hear about the details. This gave us about 1,000 active compounds from about 300,000. So we've screened about 1.2 million compounds total, and globally, the whole community, including Glaxo and Novartis, have screened roughly 6 million compounds. We think this represents essentially the entire actionable chemical universe for starting projects like this. So based on those first 1,000 hits, we then started to narrow down in the way I described strategically So we required them to be potent, we required them to work on multiple drug-resistant strains, and we required them not to affect mammalian cells. So we set these bars pretty low. 2 or 3 micromolar is not real stringent potency requirement. 10 nanomolar would be a very stringent potency requirement. 10-fold selectivity index is not anything anybody's going to write home about. So I will tell you the most interesting thing for me that came out of this work. It turns out if you have a 10-fold selectivity against mammalian cells, you have 1,000-fold selectivity for everything other than anti-cancer drugs or mechanistically related compounds. So things that hit pyrimidine metabolism, things that hit mitochondrial function work across the eukaryotic spectra. But there are many, many, many things that are specific to species. And in this case, very specific to malaria relative to mammals or even other eukaryotic pathogens that are closely related genetically. So that's a duh moment for me, right? Physiology is different, sensitivity is going to be different. The genetics being the same sets the predisposition, but not the actual effect. There, that's my precision medicine part of the talk. Genetics does not define phenotype. So based on this kind of narrowing, we went down from about 1,000 to about 500 compounds. So don't worry about the chemistry. If you're not a chemist, if you are a chemist, there won't be nearly enough to keep you engaged. Out of that work came this series, which is what I'm going to spend the rest of the time talking about. We call these the dihydroisoquinolones or the DHIQs based on this central scaffold. And what I've done here is put the set that was in the screening collection together structurally, and then pushed apart from one another based on differences on their periphery, and color-coded this data in 2 ways. So in a drug-resistant strain, it's color-coded by color. Dark blue is highly active in a drug-resistant strain. It's also color-coded by size in a drug-sensitive strain, and large is highly active in a drug-sensitive strain. So you're probably focusing the way most people do, which is on the large dark blue circles. That's not the most important part of this slide. The most important part is the fact there's a large range present in the relative activities, and that correlates with the structure of the molecules. So when you're starting out, being able to change the structure and change the effect is really important. If you can't do that, you can't adjust the other aspects of activity, like pharmacokinetics, like toxicology, without negatively affecting the positive effect you're seeking. So this is a property we look for very early in projects and had in this one. And just to sort of frame everything for you, this is a compound we refer to as 247, because nobody can remember 9 number compound codes. And the structure is over here. The details aren't really important, but I'm going to put everything else in the same orientation as we go through the talk. So how did this move forward? Well, one of the lessons we learned about doing this type of drug discovery is very early you want to move the compounds into in vivo models and ask what the problems are. So we started very early, within months, within a year of having these compounds in hand, to orally dose them in animal models and ask what happened. And in this case, I'm showing you 2 compounds that came from the screening collection. One of them was quite potent, so 10 nanomolar. This is 100-fold more potent than our minimum cutoff, right off the beginning of the project. No optimization done whatsoever. And a related compound, they're drawn exactly the same orientation, and you can see, I hope, the only difference is this bottom ring, that was about 5-fold weaker. But if I dose these into a mouse, this is showing you the concentration versus time curve for that dose. And the limit of quantitation is right about here, which is why this is a dotted line, because it's an inferred data. And you can see that this compound has essentially no detectable plasma exposure after giving it orally to a mouse. And this compound has a pretty good initial exposure. It's hitting roughly 10 micromolar concentrations in plasma, and then decaying off over 6 or 8 hours down to levels approaching the limit of quantitation. So this comparison was really important because correlated to the structures, it tells us 2 things. The problem is going to be pharmacokinetics, not potency, and not selectivity by inference from other data I'm not going to show you. And if you're a medicinal chemist, there are probably 4 of you in the audience. The key thing was these 2 methoxy groups on the bottom ring, which are well known to be subject to metabolism in the liver. And we would expect if we had deleted them, we would get a compound that were more stable. And indeed, that's a relationship that we see between the two in the early data set. So that says to us the kinds of changes we would make in this molecule in order to fix pharmacokinetic problems don't negatively affect potency. We can probably move these molecules forward and get this all balanced the way we want. I will point out, if you dose this compound, it does not cure disease in the animal models. All right, so this is the only real chemistry slide in the whole talk. And the only important part about this is I can buy these pieces and in a very short 3-step route, assemble them together and enable the project team to vary this position, and this position basically any way we wanted to. So we can make the majority of molecules we want to look at, understand how those affect activity, understand how they affect pharmacokinetics. And we couldn't really vary this piece very much. So if you care about the details, they've all been reported in this manuscript. What I'll do is walk you through a trajectory of how this works. So this is that compound I showed you initially. It's very potent, but no oral bioavailability. We were able to show that there was metabolism at this position to give the hydroxyl group here, and somewhere on this ring, again, to hydroxylate it. This is what we'd expect from oxidative metabolism in the liver. This is modeled in microsomes. We see the same thing in vivo. Those metabolic events lead to inactivation of the compound against malaria, and it also gets excreted more rapidly. We could also correlate poor oral absorption with this ring. It's very, very hydrophobic, so it basically makes a goo ball in the intestines, and that leads to poor absorption. So as we started to think about fixing that, we replaced this ring with one that was ionizable. This allows us to form a salt. This is much more water soluble. It absorbs much better. We started to play around with the primary structures here. So for example, replacing these methyl groups with a trifluoromethyl group removes that metabolism event. Same trick on the bottom ring. This gets us to this compound, St. Jude 733, which was a late lead. So none of these are changes that gave us an increase in potency. They were all changes that gave better oral absorption, better lifetime in circulation, and in aggregate, better oral bioavailability. So did they do the job well enough? I'm going to show you for a few slides 2 of the late leads. This is a molecule called 311-733, the main subject of the talk. They're drawn in the same orientation, so again, you can see the only difference is these 2 rings, here and here. And then they're color-coded here, so 733 data is in green, 311 data is in blue. And what I'm going to try to illustrate here is how we selected what went into the clinic moving forward in the project. So this is, again, a time concentration curve showing you plasma exposure after oral dosing. You can see we're still peaking at about 10 micromolar and decaying. We're now hitting the lower limit of quantitation somewhere around 24 hours instead of 6 to 8 hours. I'll point out these are 20 milligram per kilogram doses, as opposed to what I showed you before, which was a 300 milligram per kilogram dose. So better exposure from a dose that's 1/10 of what we were doing before. And I've drawn in here the range of potencies against all the strains of malaria we had tested in the laboratory to give you an idea that we're now well above that range for 16 to 18 hours in a mouse or a rat, both of which data are shown on here. So those improvements we made of the molecule structurally translated into a much improved oral bioavailability. Now, I'll illustrate here potency. This is now in vivo. So again, the same 2 molecules color-coded the same way. This solid line data is a humanized mouse model. So this is a NOD-SCID gamma-null mouse that's been engrafted with human erythrocytes for about a month. That means they're 85 to 90% human erythrocytes. And in that setting, we can infect the mouse with the human parasites and ask whether or not the drug is efficacious. And we get potencies there, ED90s of about 1 to 4 milligrams per kilogram for the compounds in this program. This is pretty good. It was good enough to move these up to the front of the portfolio globally when we had that data. But I'm telling you this in reverse chronological order. This is actually the data that we got initially. This is a murine malaria, again run in a mouse, now in a non-humanized mouse. This was the gold standard model when we started the project, because for drugs like chloroquine that aren't targeted therapies, it doesn't really matter which strain of malaria you're going after. Murine malarias, avian malarias, human malarias, they all respond roughly equivalently. But one of the things we learned about from doing physiological identification of hits is we often get enzymatically targeted compounds, and they often have big species variations. So we could have killed this project on this data, which is very non-exciting. And luckily, we actually ran what was a non-standard model at that time in a humanized model with a human malaria and realized the compound was quite good. So pay attention to model assumptions or they'll bite you in the posterior. So I really emphasized speed of activity when I was talking about what we desired in a compound in a drug. So this is the efficacy data rotated into the time domain. So parasitemia on this axis and time after treatment on this axis. 3 of the compounds in our series are here, very rapidly acting. And these are respectively a slow acting, medium speed, and the fastest drug that's in the clinic. So every compound that we've looked at in this series is actually faster acting than artesunate. This is one of the backbones in the ACTs. So it met that criteria that we were looking for going forward for a compound. And then this is actually the critical differentiating data for the project. So what I'm showing you here is exposure curves after oral dosing for different types of models, pharmacological models. So this is the same model I've showed you for efficacy. This is the ED90 driving dose, the exposure that you get that drives a 90% kill of the parasite in a mouse model. This is the exposure that drives a maximum speed of killing in a rodent model. And that's the number most people care about nowadays when they're thinking about defining exposure and efficacy relationships. These 2 color-coded graphs, again for 311 and 733, are showing you the daily exposure curve for a 7-day toxicology experiment where we dose every day. And we had no toxicology at this point that was actually detectable, even dosing up to 1 gram per kilogram in rodent models. So we didn't really have a differentiator. They were about equally potent. They looked about equal in terms of pharmacokinetic behavior. They looked about equal in terms of toxicology. But what was different was I could prove that 733 had about a 20-fold higher exposure than I could prove for 311. So when I went to talk to the FDA about opening an IND on these compounds, that's what they care about, the provable window of safety. That's what we project into the clinic in order to project what our therapeutic index is going to be like. So despite the fact that they were pharmacologically roughly equivalent, we chose 733 down here because of this differential. So I love to show this when there are trainees in the audience because nobody worries about dose linearity, usually until you're talking about actual clinical use. But in this project, it was the discriminator for what went forward into the clinic. And that's not terribly unusual, actually, in my experience. All right, so just to sort of recap with this molecule, these were the target parameters we were going for. This is a compound that's cidal, and it works on all the species we've tested. I didn't show you this data, trust me on that. It's rapidly acting in vivo, but I've showed you data that would probably convince you it's unlikely to be long acting in vivo without some sort of either repeated dosing or fancy expensive formulation. It does affect sexual stage activity. I did not show you this again, but it prevents gametocytes from developing normally, and this is predicted to prevent transmission blocking. It's quite potent, and it's quite safe. So we got one of the 2 out of the place where we got to bifurcate, and everything else we hit in terms of going forward for the drug indication we were going after. So this was declared as a clinical candidate and approved by Medicines for Malaria Venture, which is the de facto channel through which all malarial drugs go nowadays to move forward to the clinic. And at this point, the project team split into really 2 areas, clinical development and looking at mechanism of action. So I will attempt in the time I have available to take you through both of these, but we'll do the clinical development part first because for this audience, that's probably more important. And this has been all of the data underlying this summed up in this paper in PNAS a couple of years ago, if you want to see the details. So I mentioned that the landscape was shifting while we were going through this exercise. And one of the things that happened was we redefined the primary goal to be replacing artesunate, the A and the ACTs. And we stuck with that ideal goal still of a combination drug that would work as a single dose. But because the ACTs were failing, it became much more acceptable to have a 3-dose regimen. This is what's used for the ACTs. And we set as a tertiary goal, which means we'd love to have it, but we're not going to move water to get it there, being active in a prophylaxis combination, something we could sell for US use. And the strategy really became, because of money, we were running this out of an academic laboratory. We had to raise every dollar we needed in order to get this to work, to de-risk the compounds as rapidly as possible. So to back that jargon off for those of you who don't spend half your time talking to the pharma industry, this means we were doing everything we could do to kill it as quickly as possible with human data. The idea being you don't want to invest a single dollar more in something which isn't going to successfully go forward. And at the same time, we designed all those studies to generate ancillary data that would allow us to really define from a data-driven perspective with human data what was going to be achievable in a final clinical product. So we put a lot more data load under our phase 1 work in particular than is typical in early development in industry, because we wanted to be able to design the cheapest route to product coming out of there, and I'll walk you through that in a second. So other tactics that came out of this, the initial drug product was as simple as possible. So it's simply the HCl salt of that compound I showed you in a capsule. No fancy formulations at all, nothing to change solubility or absorption rate. We did an adaptive phase 1 design that had a safety pharmacokinetics arm, a phase 1A, and a pharmacodynamics arm, a phase 1B, and adaptations built into both of those in order for us to enable to get one IRB approval and run 2 clinical studies at 2 separate sites, but drive what got done in both of those settings based on the data as it emerged in the project. So we didn't have to go back and change the protocols in order to do whatever we were going to do. And there was a seamless trigger for the phase 1B based on safety and reaching the pharmacokinetics seen in the animal modeling. All right, so just to walk you through the de-enabling drug data, we made about 1 kilo of this material at a site in Hungary. We had it shipped to the US, to Kansas, at Catalent, where it was packaged up into 75 and 300 milligram capsules. So not only simple product, but very small number of dose stages going forward. We went back into rodents, now doing GLP work, and in this setting, again, very minimal toxicology. So it came out initially completely clean on all the rodent work that was done. Now, we had some discussion with the FDA because we're targeting 1 to 3 days. We're never going to dose longer than 3 days. We were allowed to do a 7-day safety study rather than what's more traditional in ID, which is 28 days. So this saved $750,000 for the project going forward. So we moved into the dog, and at 1 gram per kilogram, we finally actually got detectable toxicology. We saw a reversible anemia that looked like a regenerative anemia after 7 days of dosing 1 gram per kilogram. White feces, because poor absorption at that level, this is a huge dose. But you don't want to go to the FDA with no toxicology. They make you drop your starting dose in your clinical trial another tenfold, raises cost and increases time. So working back from that anemia, we were able to find methemoglobin production as being the underlying event. And once we had that, we could use that as a clinical lab and go back to the rat data and actually see the same thing at very high dose in the rat. So we had a toxicology and a mechanism of toxicology, which made us all breathe a little easier going into the clinic. The great thing about this is you can detect this with a standard clinical laboratory procedure. You can detect it tenfold lower levels than you see symptomology. So if you had to pick an adverse event to be predicting in phase 1, this is about as tolerable as it gets for your clinical people. So to put that data for you into a different kind of viewpoint, this is showing you driving AUCs. This is the AUC associated with the non-toxic dose, 30 milligrams per kilogram in the dog. The AUC associated with the maximum paratological response. And the AUC we actually saw in the first cohort in phase 1. And we were predicting between a 10 and 90-fold therapeutic index based on this initial scan of data. All right, so the phase 1 design was as follows. It was a leapfrog, safety decision drove dose escalation. We had a safety review for each cohort 7 days in, and they had 3 days to review the package or hold it, and then we could escalate. So this is designed to be quick, but based on safety. We were PK on every patient in every cohort, but that data was not used in making the escalation decisions. It could be used to stop escalation if we reached a plateau. And then we were triggering the phase 1B on the combination of a safe dose, a step above the dose that achieved the AUC we believe we needed to drive efficacy. That was 13,000 nanograms per hour milliliter. And like I said, there was PK, assay, and modeling going on in real time throughout the whole study. And so we found a vendor who could turn data around within a week and get us that data so we could use it. And the safety assessments included all the usual stuff, but a particular focus on methemoglobin and hemolysis. So we started at 75, no adverse events. This gave us about 2,500 microgram hour per liter. Up to 150, we get roughly dose linear escalation of exposure. Again, no adverse events. Same thing with 300, a good dose linearity, no adverse events. 600, same thing. At this point, we hit all 6 subjects in this cohort above the trigger AUC for efficacy. So we've now achieved what we wanted in terms of exposure in the patient population. Still no safety signal. So this triggered the first adaptation. We did a Fed study for PK now, another 6 patients. These were roughly equivalent, so no food effect with this drug product. Another step up to 900 milligrams, still no adverse events, but in this step we plateaued. In fact, it looked like we went down a little bit in total exposure. So we had written in the protocol that's an initial hold. We looked at all the safety data. We said, okay, we're good to go forward to do what probably is going to be the stopping dose. And I'll come back to that in a second. We also now triggered the adaptation over to the phase 1B. So the next dose in the phase 1A was a 1,200 milligram, and we saw we had indeed hit a plateau with the exposure to dose relationship. So this stopped the single dose escalation in the 1A because we're not getting any more effective exposure despite the dose increases. So we went into the phase 1B, and I'll show you that data in a second, but coming out of the phase 1B, we've now looped back to the second adaptation in the 1A, which is a 3-dose cohort, and that's ongoing right now. So the 1B, this is an interesting model for people in America. So actually Seattle's running it now, but what we do is we infect healthy volunteers with malaria. This is done by transfusing a small amount of erythrocytes that were harvested from a patient who was treated in a hospital in Queensland and cured. And we have those erythrocytes, which are parasitized, that are transfused into the subject. That gives them the disease. They're monitored by qPCR, and because of the careful way this is controlled, we know roughly when they're going to go symptomatic. And we actually initiate therapy 4 days before we expect them to go symptomatic. And we never let them get to a parasite load where they have symptomology. So they're always subject to rescue therapy, and we know AL works, and at this point it's worked in about 10 trials run with this source of the parasite. These are, by the way, college students in the University of Queensland, and they are compensated. So get a beer into me, I'll tell you about the funny toxicology data you get from working with college students. So there were 2 cohorts that were looked at. One of these was tuned to what we expected to be the minimal inhibitory concentration for the drug. So we're not expecting to see cures in this cohort, but this enables us to do good quality pharmacodynamic modeling. And because of the rescue arm, it's ethical to dose at this level. And then a group that we were expecting to have a strong response to the compound as a monotherapy, so 600 milligrams. And that's targeted at looking at the clearance dynamics. We do PK again on the day of dosing to see if there are any differences between an infected and an uninfected individual, and they're on constant PD monitoring. So they're inpatient for a week, and as you'll see, they were all rescued at the end of that time frame, and then they're on an ambulatory outpatient basis and checked every day afterwards. Target AUC, again, 13,000, same safety assessment setup being run here. So I'm showing you here the actual data from the 600 milligram cohort. So they were inoculated here. After about 4 days, you start to see detectable levels of parasite. So this is, again, qPCR monitoring. And you can see that it comes on pretty rapidly, and a couple of days later, we peaked up. This is the threshold, which is tenfold below where you see symptomology, and that's the trigger point for dosing. So they're treated with the compound here, single dose as a monotherapy. And then you can see the disease dynamics that go on after that's done. So all of the patients respond initially very rapidly, so it's the same slope we saw in the mouse modeling, a very rapid pharmacodynamic effect. Of the 6 who were dosed, 2 of them recrudesced within 2 days, and the remainders were good for about 4 days. So a good performance in this model is this curve, and we have that in some of the patients, but not all of them. Now, I didn't mention this, but we had already seen in the phase 1A, we had a few rapid metabolizers, and we're in the middle now. We have blood on these individuals, and so sorting out, we believe this is a genotype-driven variation in P450 activity, which is rapidly metabolizing the drug for some of the subjects. If that's true, the genotype that we have is actually prevalent in Caucasians, not in those of African descent, so it's probably not a huge deal for the project in the target area. But that was an unexpected finding. This is why we do clinical work and find things we're not expecting. But overall, this is a very nice performance for this compound. It's rapidly acting. It's got a deep kill early on in the therapy, and it extends out at the level we would expect from the number of parasites we're clearing. So all of these individuals were rescued with AL therapy wherever there's a red arrow. All right, so I'm going to wrap up that section for you. So we're in the midst now of, actually, phase 1B, the database has been locked. We're starting to prepare the reporting on this. Phase 1A is wrapping up hopefully this month in terms of clinical studies, and then we'll be finished up in terms of reporting by the end of the year. And we're moving now on to the 2 major foci in the project, which is assigning this to single dose versus 3 dose final product. We think probably 3 doses is the way we're going. So that's an artesunate replacement. And selecting what would be the other active ingredients to go into that drug product, which we're in the middle of doing right now. So in the medium term, we're pushing into phase 2. So we just got funding, and I just spent all morning negotiating the contract for this, for phase 2, A as a monotherapy. So this will be run in Peru, where we get patients who have both Vivax and falciparum. And at the same time, we're going to go back to that strategy I just showed you with whatever the combination product is. Make sure that it's safe, and then look again at the pharmacodynamics in the challenge model. So this positions us to be able to run the phase 2Bs with what we think will be the actual drug product in the patient populations that we're actually targeting, as long as I can raise money to do the developmental toxicology, which is the last piece. So if anybody wants to donate to the fund, talk to me afterwards. And then the whole strategy here is really to be able to wrap that up within the next 2 to 3 years into a phase 3 design that's ready to be a pivotal trial for registration of the molecule. I am desperately hoping that I will hand this off to some pharma company somewhere in the mid-band here, so that I can move on to other things instead of running these out of academic centers, which is sometimes tough. So I want to take a couple of minutes just to sort of wrap this back for you to mechanism, because I set the stage that way to begin. So we're obviously worried about resistance. We actively, with all the molecules we're working on in the laboratory, we select in culture to find resistant parasites. We always get these. You will always get these. There is no such thing as a molecule that will not engender resistance as a monotherapy. And this is a graph showing you representative data for strains that were selected with 733 or analogs in the series. And what it's showing you here is the change in EC50, the potency change on a log scale, versus the number of parasites we have to inoculate to see that change. So this is a crude measure of the allelic variation frequency without having to do all the complicated dynamics you got to do that study. And I've drawn on here a box which is generally accepted in the field as safe. So it means your resistance potential profile is roughly equivalent to most of the other stuff out there. And then the 2 dots outside of that, that's a tovaquone, which didn't make it through phase 1 without having resistance. Phase 1 was done in a disease-endemic country. And just little raw data for the data junkies. This is what one of these shifts looks like when we select these. So about a decade ago, when sequencing got reasonably cheap, we started with a collaborator sequencing all of these as they were made. And the blood stage is a haploid organism, so it's pretty easy to see what happens there. And an interesting thing happened. Every single clone that we selected with this molecule mapped back to a single gene, PFATP4. At the time, it was annotated as a calcium channel transporter. Its closest homolog, a 23% identity, is circa, muscle circa in mammals. We do not have any muscle toxicology, if you're worried about that. In many of the clones, this was the only non-synonymous change we saw in the genome at a 300 coverage level. So there's a very strong monotonic selection for variation in this allele. And so this starts to make you feel very comfortable that this is what's driving resistance, and possibly that this is a target. That's one of the two ways you get that kind of an effect, although in something that's labeled as a transporter, you worry about that. So while we were doing this work, the function was worked out, and this protein is actually a sodium proton antiporter that's expressed in the plasma membrane of the parasite, and it's important for adjusting ionic content post-infection in the red blood cell. So it sets up the homeostasis that lets the parasite live in that condition. And I've annotated here, this is a homology model built from a rabbit circa structure. It's the only structure I have available. And they're color-coded for the fold resistance that are caused by mutations underlying these side chains. And what got really attractive about this is it's probably pretty obvious. They're all clustered together. They're in the channel portion of the transporter, the transmembrane portion. And the ones that are really high fold resistance are even closer together in something that looks like it might be a small molecule binding site. Now, we were never able reliably to get resistant clones when we tried to select them in vivo. One of our collaborators spent almost 2 years trying to do this. We got one resistant clone. It was only two-fold resistant, and it was not stable. So hold that in your mind. So because we had a predicted function, and I had coincidentally met the person who had actually done the electrochemistry on this, electrophysiology on this, we were able to ship the molecule and an inactive version of it, as well as some of our resistant clones over to his lab. And he could show changes in sodium flux and in resting sodium levels before and after treatment with the molecule that were really congruent with what we expected based on his predicted function. So when we dump this molecule on top of the parasites, the sodium goes up. It goes up from 10 millimolar resting level to 120 or so. It does that pretty rapidly within minutes of treatment. If you use a resistant clone, it's much more resistant to that change. And if you use an inactive variation on the molecule, same story. So at that level, everything's looking pretty good. Nobody understands functionally why this would lead to death of the parasite. So I wanted to show you this last little bit of data. This is showing you on the right a treated parasitized erythrocyte. We'll see if I can. So these are actually time-lapse video microscopy. This one's been running the whole time the slide's been up. I just started this one. So in a normal parasitized erythrocyte, you still have the discoid appearance of the erythrocyte. Post-paratization, it becomes knobby instead of smooth. Right? And you can see here the parasite is moving around inside the erythrocyte quite actively. And in fact, it's dividing. We could sit here and watch that if I gave you a longer time frame of this. So parasite activity, discoid shape, knobby surface. As soon as we treat this, in fact, as rapidly as we can refocus, we have a spherical, dense, smooth erythrocyte and a dead as a doornail, non-motile parasite, which is non-dividing. Now, this is not actually dead at the level of being killed. It's simply hanging out. So if we wait in the laboratory, it'll take 96 hours or so for it to slowly die off. It just isn't happy in this state. Any hematologists in the audience? They're asleep already. See, I had one hematology slide for you and you blanked out already. So this has a really interesting appearance. It looks like a 57-day-old erythrocyte. It looks like an erythrocyte that's going to get cleared. Right? And that's exactly what happens. So the driver for the in vivo rapid speed of effect is macrophage clearance solely of parasitized treated erythrocytes. It's a host-mediated clearance mechanism. We think, waving my hands around in the air, that this is what's driving the difficulty in getting resistance to these compounds in vivo, because your body's working really hard to get rid of aged erythrocytes. It perceives these as in that state and gets rid of them. So we think what's really going on here is the mutation, the PFADB4 is the primary target. The primary effect of that is changing the sodium state and flux in the parasitized erythrocyte. That leads to this morphology, which is congruent with an eruptotic phenotype in the erythrocyte, and that leads to in vivo clearance of the parasite. All right, I managed to actually get that done. Awesome. So just to wrap this talk up for you, so what do we get out of this? Well, if you're one of those people who would argue with me that you can't do translation in academia, I doubt there are too many in this audience, but I always have at least one. I hope I've demonstrated to you that you can go from a cocktail napkin to the clinic in an academic environment with academic collaborators, with funding that's available to academics. And by the way, this costs less than $5 million total. Now that we're moving into phase 2, it'll be another 4. We will be through phase 3 with less than $50 million entire total investment. So you can do this. You need a core team that's going to drive it forward. I'm the cheerleader. That's my function. You need good people in the laboratory who are going to keep institutional knowledge and make it work. And I think the beauty of doing it in this environment is your R01 and similarly funded science deals with the failure rate. You don't write that into your cost like you do if you're a for-profit entity. If you're doing good science, you're serving that need, but you're moving the project forward at the same time. People miss that part of it. The R&D encumbrance is much less to an academic because it's spread out in time, averaged, and covered by the government. I hope I've convinced you that successfully translating requires new science. I tried to highlight the spots in the talk where the current state-of-the-art model didn't work, where we needed to understand things we didn't understand, where we couldn't predict. We had to be good scientists to make this work. And I hope I've convinced you that I'm a benign dictator, especially those faculty who live in my college. The project management made this work. Me paying attention to it made it work. But I collaborate with people all over the world. We don't pervert what they do in their own academic labs. We don't force them to do experiments. We convince them. We enable their own work by bringing this project to hang everything on. And these are great opportunities for students and trainees. They're out at the bloody edge of the spear, learning how to do things in an environment where it's hard. And they have to work well and work with others in order to make things work. And I'll leave you with that message and all the people who actually did all this work I talked about and the people who paid for it in the bottom left corner. And I'm happy to take questions.